Nothing in life is to be feared. It is only to be understood. Now is the time to understand more, so that we may fear less.
Hi there. I'm Alex. I know you already want this COVID-19 pandemic to end now. But since there is still no available vaccine or cure for it yet, please stay at home and stay tuned on this video to learn more. You might just learn something that will make you become the next person who'll find a cure for COVID-19. Enough with the chat, let's move on with our topic entitled Natural Products in Modern Drug Discovery and Its Role Against COVID-19. This presentation is divided into two parts. The first part will focus on the role of natural products in modern drug discovery, while the second part will give an emphasis on the role of natural products against COVID-19. So what are natural products? A natural product is a chemical compound or substance produced by a naturally occurring living organism such as plants, animals, and microorganisms. It can be further classified as a primary or a secondary metabolite. However, the latter is usually more associated with the term. Natural products have pharmacological properties which may contribute to vital drug discovery and design. Since ancient times, natural products have been widely used to treat many diseases. Medicinal plants and microorganisms were the major source of medicines over many centuries. There are many examples of plant-derived extracts or compounds isolated from plants that have been widely used in the treatment of many significant diseases. To name just two examples, poppy and marijuana have been used as early as 4,000 years ago. However, it was only in 1806 that Friedrich Saturner isolated the alkaloid called morphine from poppy. This was an event that prompted a continuing search for other plant-derived medicines. Then, in 1824, Pierre-Jean Robiquet isolated the antitussive agent codeine, also from poppy. And finally in 1848, George Merck isolated the antispasmodic alkaloid papaverine from the same plant. Amongst the many other important active principles isolated from medicinal plants are caffeine, obtained by Friedleb Ferdinand Runge in 1820 from Coffea arabica. Atropine, a muscarinic antagonist, isolated from Atropa belladonna by Heinrich F. G. Mein in 1831. Digoxin, a cardiac glycoside, isolated by Claude Adolphe Nativelle in 1869 from Digitalis lanata. Tubocurarine, a toxic alkaloid historically known for its use as an arrow poison but is now used as a muscle relaxant. It was isolated by Winster Steiner and Dutcher in 1943 from the South American plant Conradendron tomentosum. The annual global medicine market is worth around $1.1 trillion. About 38% of these medicines originated directly or indirectly from natural products including plants, at 25%, microorganisms, at 13%, and animals, at about 3%. Natural products constitute an extremely important resource for global pharmaceutical companies working on the development of new medicines. They are used as a direct source of therapeutic agents, both as pure drugs and phytomedicines, a source of raw material for the development of complex or semi-synthetic drugs, prototypes for design of lead molecules, and as taxonomic markers for discovery of new drugs. About 1/3 of the best-selling drugs in the world are natural products or their derivatives. Between 1981 and 2014, the US FDA approved 1,562 drugs. 4% were unaltered natural products, 9.1% were botanical drugs or mixtures, 21% were natural product derivatives and 4% were synthetic drugs but with natural products pharmacophore. There are many examples of globally best-selling medicines that originated from natural products, notably from higher plants, microorganisms and animals. Some good examples are the anticholesterolemic agents, simvastatin, lovastatin, and atorvastatin, pravastatin. The antihypertensive agents, captopril and enalapril. The immunosuppressive agents, tacrolimus, rampamycin, and cyclosporin A. The antibiotic and antifungal agents, erythromycin, amphotericin B, penicillin, and clarithromycin. Now, we have reached the second half of the presentation. The big question still remains: how are natural products able to fight COVID-19? A virus is a unique pathogen which is incapable of replicating without a host cell. It utilizes the host cell environment and cellular factors for its propagation. This unique feature of viruses makes it difficult to design a treatment to attack the virus or its replication directly without any adverse effects on the infected cells. In this regard, viral diseases caused by pathogenic virus infections are still the leading cause of death in humans worldwide. Although effective vaccines have led or might lead to the eradication of important viral pathogens, such as smallpox, polio, and mumps, other viral diseases, such as the HIV, hepatitis C, and more recently the COVID-19 have been proven difficult to combat. Knowledge of the intrinsic characteristics of a virus is critical for antiviral treatment development. With awareness of its molecular structure, inoculation, and replication mechanism, it is possible to develop drugs from natural products that can specifically combat the virus without severe adverse effects to the host. Now, let's talk about coronaviruses. Coronaviruses are large, enveloped viruses, having a spherical morphology, although they can often be pleomorphic, with a single-stranded RNA genome of positive polarity. Coronaviruses also have prominent glycoprotein spikes. It can infect humans and a wide range of animal species. The Coronaviridae family is classified into two subfamilies, Letavirinae and Orthocoronaviridae. The subfamily Orthocoronaviridae includes alpha corona, beta corona, gamma corona, and delta coronaviruses. Of the four genera, the first two infect only mammals while the last two primarily infect birds. 7 types of coronaviruses infect humans, the first 4 are agents of seasonal infections. The last 3 coronaviruses belong to the order nidovirales and are SARS agents, causing infections with a severe clinical picture and high mortality. These 3 coronaviruses are SARS-CoV-1, MERS-CoV, and SARS-CoV-2. OC43, HKU1 and all the 3 novel SARS coronaviruses are beta coronaviruses. Coronavirus disease 2019 or COVID-19 is an infectious disease caused by severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. It was first identified in December 2019 in Wuhan, Hubei, China, and has resulted in an ongoing pandemic. As of the 7th of October 2020, around 36 million cases had been confirmed worldwide, and more than 1.05 million deaths had been attributed to COVID-19. Common symptoms of COVID-19 include fever, cough, fatigue, shortness of breath or breathing difficulties, and loss of smell and taste. While most people have mild symptoms, some people develop acute respiratory distress syndrome possibly precipitated by cytokine storm, multi-organ failure, septic shock, and blood clots. After knowing the virus, how are we going to fight or treat it? Since the 2003 to 2004 SARS outbreak, several natural products from flora and fauna have been tested in terms of their anti-SARS-CoV-1 activity and was used as a scaffold in drug development. These natural metabolites include flavones, flavonols, fatty acids, tannins, terpenes, and alkaloids. The diversity of these chemical classes is related to the different mechanism used to inhibit the coronavirus. Chemical structures of these natural metabolites present common features that corroborate the conclusions of a group of researchers, who, through in silico analysis, showed that SARS-CoV-1 inhibition requires chemical structures containing a hydrophobic aromatic ring, hydroxyl groups, and carbohydrates moieties. In the ongoing COVID-19 pandemic caused by SARS-CoV-2, natural products have a huge potential in coronavirus treatment, as can be observed in the history of SARS-CoV-1 combat. This is attributed to genomic sequence of SARS-CoV-2 closely resembling SARS-CoV-1 at 79.5%. A more thorough understanding of the genomic sequence of SARS-CoV-2 enabled the identification of the critical protein or enzymes involved in its inoculation and replication processes. The genomic data demonstrated that SARS-CoV-2 inoculation and viral replication occur mainly through three major proteins or enzymes. These proteins or enzymes are the papain-like protease ACE2, spike protein TMPRSS2, and the 3-chymotrypsin-like protease 3CL-Pro. Among those three structures, the ACE2 and TMPRSS2 are part of the host cell. The ACE2 is an analog of the angiotensin-converting enzyme type 1 or ACE and part of the renin-angiotensin system responsible for blood pressure regulation. Most of anti-COVID-19 developments have been focused on the ACE2 inhibition, although acting on such a central pressure control system is probably not the best approach. The spike protein with which SARS-CoV-2 interacts is the transmembrane protease serine type 2, TMPRSS2. It was estimated that after SARS-CoV-2 binding with ACE2, host TMPRSS2 promotes virus fusion with the infected cell and its activation by cleavage of virus protein. Lastly, the 3CL-Pro is responsible for the proteolytic cleavage of virus polypeptide in 11 non-structural proteins responsible for its replication. 3CL-Pro's occurrence only within SARS-CoV-2 and not in the host cell have dragged attention as a possible inhibition site for COVID-19 treatment development. ACE2 inhibitors present amphiphilic molecular structures usually with an aromatic moiety in order to enable their interaction with the protein, a similar pattern observed with SARS-CoV-1 inhibitors. Pharmacophore analysis of suggested natural metabolites capable of inhibiting SARS-CoV-2 through ACE2 interaction usually have structural features in accordance with Lipinski's rule with molecular weight lower than 500 Daltons, less than 5 hydrogen bond donors, and a log P under 5. The number of hydrogen bond acceptors was more variable among suggested phytochemicals against SARS-CoV-2, ranging until 20. The first in silico screening for anti-SARS-CoV-2 natural metabolites was within traditional Chinese herbs, such as the species of the citrus genus. The virtual molecular docking of several metabolites such as naringin, naringenin, nobiletin with ACE2 have resulted to a binding energy of -6.85, -6.05, and -5.42 kilocalories per mole, respectively. The lower the binding energy, the greater is the potential of a metabolite to inhibit the target protein or enzyme. Similarly, virtual molecular docking of some flavonoids such as hesperitin, hesperidin, and neohesperidin with ACE2 have resulted to a binding energy of -6.09, -4.21, and -3.78 kilocalories per mole, respectively. In another study, it was indicated that glycosylated derivatives of quercetin, such as quercetin 3-glucuronide 7-glucoside and quercetin 3-visionoside, could also present promising inhibition towards ACE2 with binding energies around -8.3 kilocalories per mole. These flavonoids can be found in Indian long pepper, turmeric, and wormwood. So far, at the end of May 2020, among the studied flavonoids, psilocybin have been suggested to have the lowest binding energy towards ACE2. Psilocybin is a flavolignin and one of the major metabolites obtained from milk thistle seeds, a plant with traditional usage in chemoprevention, anti-inflammation, and in the treatment of digestive disorders. Research for TMPRSS2 inhibitors is the lowest among the major replication proteins despite the fact that it already known for its involvement in the inoculation and replication of influenza virus, cancer, and the SARS-CoV-1. TMPRSS2 inhibitors require similar structural features as those previously described for ACE2 inhibitors, presence of hydroxyl moieties for hydrogen binding and presence of aromatic rings. Identified TMPRSS2 natural inhibitors include flavonoids, terpenes and peptides. Through in silico analysis, bakelin have been reported to have a binding energy of negative 8.46 kilocalories per mole towards TMPRSS2. The major natural sources of bakelin are the Scutellaria and Oroxylum genera. This flavonoid have been reported to have therapeutic, neuroprotective, antioxidant, anti-inflammatory, renal protector, and anti-cancer properties. In addition, this flavonoid also presented remarkable activity as inhibitor of the Zika virus. In a separate study, a very great inhibition potential towards TMPRSS2 was seen on geniparside, an iridoid found in the Gardenia genus and endemic in Central America and China. The inhibition of 3CL-Pro, the main protein of SARS-CoV-2, has received more attention within researchers as it could prevent virus inoculation on the host. Although the 3CL-Pro is an enzyme specific to the virus, the one within SARS-CoV-2 has a large structural similarity with the one present in SARS-CoV-1 at 96.08%. Through molecular docking of natural metabolites, promising results in inhibiting 3CL-Pro were found for camphorol, quercetin, luteolin 7-glucoside, dimethoxycurcumin, naringenin, apigenin 7-glucoside, catechin, epigallocatechin, and oleuropein. In addition to the promising results observed to flavonoids, volatile terpenoids are also specialized metabolites that present some very interesting preliminary results to indicate possible use of these substances against SARS-CoV-2. In this case, the already existing supply chains from industries that produce essential oils increase the sustainability of this kind of prospection. The mono and sesquiterpenes geraniol, linalool, beta-farnesene, and nerolidol presented in silico inhibition of 3CL-Pro with binding energy of negative 24.71, negative 24.05, negative 27.56 and negative 26.44 kilocalories per mole, respectively. These terpenoids can be found in several plant species with ancient and very well-known uses in food, medicine, and aromatherapy. This include the lemon balm, lemongrass, lavender, geranium, basil, mandarin, cinnamon, chamomile, ginger, and copaiba among others. Although the development of bioactive natural products against a specific disease, such as COVID-19, is faster than vaccine development, it is still an arduous task due to the diversity of natural metabolites and their chemical complexity. It is noteworthy to mention that virtual screening for bioactive compounds through molecular docking has been a useful tool in natural products research to shorten the time spent in phytochemical screening of several extracts. In the face of this great global challenge, we are striving for a COVID-19 treatment that can be quickly produced and easily distributed. Natural products could provide an answer to this dilemma, as they often have low toxicity and some have already been used in the pharmaceutical industry for their bioactivity, including antiviral activity. The similarity between SARS-CoV-1 and SARS-CoV-2 also brings light to the development of new drugs or even vaccine. Great hope has emerged from the possible anti-SARS-CoV-2 activity of flavonols, flavonones, and flavonols and the fact that these metabolites have a large occurrence within angiosperm plants. As most of the present research is theoretical or does not present analytical validation, a long path is still ahead in terms of biological analysis, optimized extraction, and production. Thank you very much for watching, and have a great day.